Why the electrons? I'm not hearing you well.
Yeah, the previous slide. Why does the electron flow when ATP synthesis drop? That one.
Yeah. So this slide. So why does the electron flow stop? See, let me go to the cartoon. Yeah. So the protons in the intermembrane space can return to the matrix only via this ATP synthase pump. So if this is blocked, that is, if you block ATP synthesis, then the protons don't return. So now the energy available from the electron flow can only create a certain a difference in the concentration between these two, beyond which what happens is the energy required to push any further protons across this will be more than what you get from the electron flow, because they both reach an equilibrium state. So energy available from the electron flow equals the energy required to keep this concentration gradient. So unless otherwise you allow protons to come through, there is no pumping that is going to happen. And that is why these systems, like when these are not going to pump, then the electron flow is also not going to happen, because the activity is dependent on the conformation changes on these complexes. Like for example, the electron flow through complex 1 changes its conformation such that pumps the proton across. Now if the proton pumping is not possible, then it is going to remain in a locked position and it's not going to allow electron flow. So that is the mechanistic detail, but from an energy perspective, the energy available through this is at equilibrium with the energy required to maintain that high concentration gradient of protons. So is this clear?
Okay, thank you.
You got it or no? I can give it another attempt if it's not clear. So the main thing is these proteins are going to get locked in a conformation. So unless otherwise proton can be pumped across, they are not going to have a conformation that will allow the electrons to flow through. Okay, great. So let us move on to today's class. Okay, let's continue on the photosynthesis theme that we started yesterday. So this is the place where it all happens. Okay, so chloroplast. So this is like mitochondria, another organelle. So the mitochondria is shown adjacent to this big chloroplast. So both of these were independent prokaryotes long time ago. So this is an endosymbiotic relationship between eukaryotic cells and these two prokaryotes. So even at the cellular level, the most fundamental unit of your existence, you are not an individual. Okay, so that should be a humbling understanding of the facts. So even at the most fundamental unit, you are in coexistence. You are sharing with someone else. That is mitochondria if you are animal, and if you are a plant, then you have chloroplast and mitochondria as well. They have their own DNA also. So anyway, the photosynthesis happens in this structure called the chloroplast. So let us get a closer view of this chloroplast. So chloroplast again has an outer membrane and inner membrane, and this intermembrane space is not critical for us for the photosynthesis part. And for that, the mitochondrial intermembrane space equivalent is the space in this flattened discs. Okay, so each one of these discs is a thylakoid, and a stack of the thylakoid is a granum. Okay, so a stack of thylakoid are granum, and you have multiple grana in a chloroplast. And the protons are going to be pumped into that inner space of the thylakoid, and the machinery required for carbon assimilation reaction, they are going to be in the open inner space of the chloroplast, which is called stroma. Okay, not matrix as we saw in mitochondria. So this thylakoid, inner space of the thylakoid is where the proton gradient is going to be built. That is where the pH is going to be very, very low. So you're going to have a lot of protons there when photosynthesis happens. And it is going to flow across the membrane into stroma, this whole open space there. And that is the concentration gradient change via ATP synthase pump. And this is a transmission electron micrograph of an actual chloroplast. Okay, so before we go ahead with photosynthesis, we already saw how the relationship between the heterotrophs like animals and the autotrophs like the plants and some of the bacteria like cyanobacteria. So you have a cycling of matter, right? Carbon, oxygen, and hydrogen. So that is where we started with, and now what we are going to do is we are going to refresh very, very elementary physics about light. Okay, basic properties of light, essentially light absorption alone. So most of you have learned this multiple times by now in school and probably in your chemistry and physics courses here. So a photon is a quantum of light, and when a molecule absorbs a photon, an electron gets lifted to a higher energy orbit, and we call that excited state. And this is an unstable state, and it readily comes back to its normal orbital, and that is ground state. And during this returning trip, the energy absorbed is dissipated in the form of light. Usually that is at a higher wavelength than the wavelength at which the original absorption happened, and that we call fluorescence. And sometimes it is heat, simply the molecular motion is the cause the heat, or sometimes this may be used to do a chemical work. So this is what normally happens when a chromophore, that is a molecule that can absorb light, undergoes when you have when you shine light on it. So remember these two terms, excited state and ground state. So the part of this principle that is really important for photosynthesis is given here. Here what actually happens is, instead of that energy dissipating in the form of light or heat, here that is a part of that is transferred to an adjacent molecule. Okay, direct transfer of excitation energy from one molecule to a neighboring molecule. That is what is crucial for photosynthesis. And that quantum of energy that is transferred from one excited molecule to a neighboring molecule is what we call exciton. And the transfer of that we call as exciton transfer. So this word exciton and exciton transfer are key for photosynthesis. Okay. So this is not the quantum of energy that lifted electron to a higher orbit. This is the quantum of energy that is transferred from such an excited molecule to a neighboring molecule. That is what is exciton. So clearly remember the difference between exciton and photon. And the process of transfer is exciton transfer. Here the energy is not dissipated in the form of fluorescence or molecular motion or in doing any other chemical work. So what are these chromophores that are responsible for photosynthesis, and that is what we are going to look at here. So this structure again is a conjugated double bond structure, a ring-like structure. It's very similar to the porphyrin ring of heme moiety that we encountered in cytochromes in the in complex 3 and 4, and then cytochrome C that is on the inner surface of the inner mitochondrial membrane. So the only primary difference is here you have a magnesium instead of ion that is coordinated to the nitrogen atoms. Okay. And so you have certain variations here, and the variations are only in the side chain and they are marked here. So chlorophyll A is this basic structure. All of them have this phytol isoprene structure, which we have encountered multiple times when we learned the lipid soluble vitamins. So this is chlorophyll A, and in bacteriochlorophyll, which is present in purple bacteria, green sulfur bacteria, etc. We will learn about them as we move along. So they, instead of the regular chlorophyll, they have what is called a bacteriochlorophyll. So there, instead of this, they have this structure. Okay, so that's the difference. And chlorophyll B has an aldehyde group instead of this methyl group. And this bacteriochlorophyll, in addition, has saturated bond between these two carbons here. Okay, and without these modifications, it is chlorophyll A. So this chlorophyll A and chlorophyll B are the main chromophores in the plant and cyanobacterial light absorption system. Okay, in their light absorbing unit, which we'll learn in few minutes. And in more ancient bacteria like purple bacteria and green sulfur bacteria, they have this bacteriochlorophyll. So they have very important differences in terms of the way they do photosynthesis that will come in a couple of slides. Okay, so and the chlorophyll molecule actually a very important concept here again, so pay attention closely and remember it well. So the free chlorophyll, if you excite with light, the energy absorbed quickly is dissipated and it comes back to ground state with no exciton transfer or anything. So that's what happens when you have free chlorophyll in solution. But on the other hand, in the thylakoid membranes, the chlorophyll molecules as well as other pigments that we are going to learn, they are all part of protein structures, and they are all oriented in very specific way such that before the energy can dissipate, it gets transferred to an adjacent molecule. So much of all this transfers happen in solid state, and that's very critical for transducing the sun's energy into useful chemical form in photosynthesis. So that's the main point here. The chlorophyll is always associated with specific binding proteins, and such a structure of these chromophores and proteins and everything put together we call as light harvesting complexes. Okay, this shows the structure of one such LHC, in which chlorophyll molecules are fixed in relation to each other and to other protein complexes and to the membrane. They are in very fixed orientation with no molecular motion or diffusion possible. And this is what enables transfer of that energy, exciton energy, in about 3 picoseconds from the time of absorbance. 10 picoseconds is what is required for it to come back to ground state without transferring to any other molecule, like purely by dissipation. And this transfer, due to all these tight orientations, it happens faster than the 10 picoseconds. So that is the primary thing achieved by this arrangement of multi-protein, chlorophyll and other pigment complexes. Okay, so the other important point we want to consider here are the absorption spectra of these chromophores. So if you look at it here, let's look at chlorophyll B. So it has an absorbance. So the x-axis is the wavelength. So you have around this region a peak absorbance, and then it has another peak. At the other extreme end, around around the red area, you have another peak. And chlorophyll A broadens that absorption area. It absorbs beyond that in this extreme, and in the other extreme also it absorbs at shorter wavelengths. Okay, so therefore they cover, between the two of them, they cover a broader area than what either one of them can do. In addition, we have accessory pigments, which we learn in a minute. Beta carotene, for example. Remember beta carotene, we have learned when we were learning about the vitamin A. So it is it's basically two vitamin A molecules put together is a beta carotene. So that helps in absorption in photosystems, and it has an absorption spectrum shown here. And lutein is another one that is found in chloroplasts. That molecule has this absorption spectrum. While these are in bacteria. Okay, so this is not in the same chloroplast. So don't assume that between the various one we are covering this entire sun's spectrum. Okay, so in plant chloroplasts, then this region is primarily not covered. Okay, so this is one more important concept. So the photosystem is basically an array on the thylakoid membrane. So the blue things you see are all the lipid bilayer, the head groups of the phospholipids. So now you realize the importance of the chemistry of biomolecules that we learned. So that is what makes us understand what is the head group, what is the tail group, all that. This phospholipid, okay, this membrane. On that, you have these arrayed, this green structure. So this is equivalent of this. Okay, so you have protein molecules. I didn't get into the details. So a functional unit is a trimer with 36 chlorophyll, 6 lutein. So this yellow ones are the lutein. So then you have chlorophyll A 7 of them. And then chlorophyll B, 1, 2, 3, 4, 5 of them. So this whole thing is one unit, one light harvesting complex. Okay, so it has one kind of accessory pigment, but it has 6 of them. And then you have the chlorophyll A and B, and then this protein. So like this, a trimer is what is one LHC complex. And that is each one of these green ones here. Okay, and these orange ones have no chlorophyll. Instead they have accessory pigments, and when light is absorbed by one of them, the energy from that excites a neighboring one, and so on from this entire antenna. So this whole thing is an antenna, light harvesting antenna. From that, finally it is transferred to a central chlorophyll molecule, very much like these ones, but these have special chlorophyll A and chlorophyll B. These are called the reaction centers. Okay, photochemical reaction center. So this is where the energy absorbed from the light is used to make one electron donor to donate an electron, and an electron acceptor to accept an electron, and you therefore create a charge gradient. Okay, so an electron is really lifted to a molecule with very low reduction potential, and from there now it can do a downhill transfer, like what happened in the mitochondrial inner membrane. So that electron transfer, actual electron transfer, and therefore generation of a charge gradient happens in the reaction center. So from these, it's just the energy transferred, and their electrons go back to normal. And finally, it all comes to this. So this entire array on the thylakoid membrane. So if you take a thylakoid membrane, a part of it, and there you have so many of these, and all of them harvest energy from the sun, and it is transferred to this reaction center. Okay, so antenna chlorophyll bound to protein, carotenoid and accessory pigments on the membrane. So these molecules absorb light energy, transferring it between molecules until it reaches the reaction center. Okay, so reaction center is where the energy of a photon into separation of charge and initiating the electron flow. So this is an important thing you need to remember. So chlorophyll molecules, first of all, do not exist in isolation. They are part of a rigid protein pigment association called LHCs. And these LHCs, many of them exist on the thylakoid membrane, calling the light harvesting antenna. And from them, it gets transferred to the reaction center. So at the reaction center, now let us look at this. So in each one of these antenna molecules, antenna chlorophyll molecules, like one of the many green structures that we saw. See, these are inserted through the membrane because these are integral membrane proteins. Okay, so they have hydrophobic residues which can interact with the membrane's hydrophobic tails, lipid tails. So in any one of these antenna molecules, light absorption leads to the electron excited to a higher energy level, and then it returns to the normal by transferring to another adjacent molecule, and so on it goes on, and then finally the energy is transferred to the reaction center. And when that happens, so this central part is actually the reaction center. So this is an acceptor, this is a donor. And here it is lifted, and therefore it is really donated to a molecule that can accept electrons. And when that happens, you have a gap, and this is called electron hole. Okay, and that needs to be filled, and that comes from an electron donor. So the ultimate electron donor in cyanobacteria as well as in plant chloroplasts is water. So water is split, oxygen evolves, and the two hydrogen atoms, and they are the ones that provide the electrons to this. Okay, so in that we will learn towards the end of this discussion on the photosynthesis. I'm not sure we'll cover it today. We may not go that far. So that we will see later. So this is right now. For our conceptual understanding, it is an electron donor. So from electron donor, this gap is going to be filled. This electron hole, so written here, the electron hole in the reaction center is filled by an electron from an electron donor. So this is what happens. So now at the end, what you have is a clear separation of charge. An acceptor has an extra electron and a donor does not have an electron. So you have a charge difference. And this is what initiates the electron flow. So much of what we understand comes from studies in bacteria because they are simpler systems, easy to grow in large number and isolate, purify, crystallize, and understand the mechanisms of electron transfer. So most of the work has been done using bacterial systems. A variant in bacteria that does photosynthesis is purple bacteria. Okay, so this purple bacteria uses bacteriochlorophyll. Okay, so the bacterial cells that use bacteriochlorophyll, they do not split water. So water is not their electron donor. So therefore they do not evolve oxygen. They do not produce oxygen. So the oxygen produced by photosynthesis comes from water, splitting water. It does not come from carbon dioxide. So remember that. Often people confuse that. So therefore, these are non-oxygen evolving photosystems. They use the sun's energy. So photosynthesis happens. So do not always connect photosynthesis to oxygen or water. That is only with cyanobacteria and plant chloroplasts. So in purple bacteria, what actually happens? Let us look at it, because this is where much of the study has been done. So the reaction center, when gets excited by the exciton transfer, so this is the excited state. So the energy difference is shown in the y-axis. So from there, electron flows to a pheophytin, which is a quinone like hydrophobic pigment, light electron carrier, intermediate electron carrier. So this pheophytin, so we didn't consider pheophytin here because I just wanted to keep it simple, and so we learned about chlorophyll A. Oh, I skipped this carotenoids, but I can't skip. Yeah, so pheophytin we don't have. But a book has it. I just omitted some of the structures from the bacteria because that you need not learn all of that. So beta carotene is important because these accessory pigments called carotenoids extend the range of light absorption by chlorophyll A and B. So they also transfer it to the reaction center. So this is the structure of beta carotene. This we are seeing for the second time. We saw this when we learned about vitamin A. You know, this all trans retinol and 11 cis retinol and retinoic acid, all that we learned, and that's precursor is beta carotene. And this is the structure of lutein. So these molecules also help in light harvesting. So instead of those kind of molecules, you have pheophytin in this purple bacteria. So that is the one first accepts the electron from the reaction center, and then from there to plastoquinone. So this is like ubiquinone like molecule. From there to cytochrome BC1 complex. This is more like the mitochondrial complex 3, which is transferring electrons from ubiquinol, that is the fully reduced ubiquinol QH2 to cytochrome C. This that complex probably evolved from this. This is a more ancient structure similar to the cytochrome, so the complex 3. And from there, it flows to cytochrome C, very much like what happens in complex 3 in mitochondria. And from there, it goes back to reaction center. So the excited electron actually goes around and comes back. So there isn't actually an independent electron donor. The same thing returns back here. In chloroplasts and in cyanobacteria, a water molecule would have been split and the electron from that would have come to fill this gap in the photosystem. Whereas in purple bacteria, it is the same thing. But this downward grade flow, that energy released from that is used to pump protons. Just like complex 3 in mitochondria, this pumps protons across the membrane, bacterial cell membrane to periplasmic space. And from there, when the proton comes via ATP synthesis, it makes ATP. So that is how this bacteria harvests energy from sun's light. Okay. So it uses the spelling for pheophytin, quinone, they are all here. So here there is no oxygen evolving. So remember this, do not always associate photosynthesis with splitting water. So that happened later, primarily because when these bacteria are originally growing in an aqueous, like marine environment, the abundant molecule available there is water, although it is a poor electron donor. And when they evolved a mechanism where they could use water, they switched to water. And that is how water became the electron donor in later organisms, in cyanobacteria and so on. And that allowed them to spread into the ocean. They need not be in a specific place. Like for example, the other bacteria we are going to learn, the next system uses H2S, hydrogen sulfide. So the hydrogen sulfide is available in only deep sea vents, like hydrothermal vents that exist, and there only you have H2S, and these bacteria can grow only there. But the moment they switched from H2S to H2O, then H2O is a ocean full of H2O. So literally, so then they can spread around. And that is how oxygen started accumulating in the atmosphere. Okay, so oxygen did not exist in the atmosphere before photosynthesis happened. So all the oxygen in the atmosphere came from splitting water and evolving oxygen. So the next one that we are going to see is a slight variation in this theme. Okay. Okay. Before going into it, we need to understand this structure of this photosystem. So that is shown here. Okay. So this has, so the main structure, so this is the cytochrome C part. So ignoring this yellow cytochrome C having 4 heme groups. So the heme alone are shown here with the electron flow shown by the blue arrows without the protein side chain. So this has the protein side chain. So within the membrane, you have 3 of the polypeptides. So the gray, and then you have the blue shown here, and then this orange color. Okay. So among them are oriented. So this, the green structure is actually the heme moiety, the chlorophyll, bacteriochlorophyll. Okay, so this bacteriochlorophyll is distinct from chlorophyll A and chlorophyll B in that the bacteriochlorophyll containing systems do not use water as the electron donor. So these pigment molecules are associated with these proteins, like the blue one, orange one, and the gray one. So 3 of them. So here you have the electron flow diagram along the molecule. So you have the cytochrome C. Then so the number here indicates the time taken for the transfer. This is 270 nanoseconds from cytochrome C to this chlorophyll, bacteriochlorophyll. Okay, so this is a special pair. So this is the reaction center. So then you have accessory pigment chlorophyll, bacteriochlorophyll. So 2 pairs, so 4 molecules of that. Then so that is 3 picoseconds. Okay, so when the light excites, for this to go to the next one is only 3 picoseconds. Then by the time it comes here, it is 200 picoseconds. Then very quickly it goes to this, and then finally from this quinone, it is going to go to the cytochrome C, and then finally to this. So let us go through that again. So this is the starting point. Okay, so light on the reaction center. Then to the next one, pheophytin, and from pheophytin to QA, and then via this ion you have the QB, and then from this to the cytochrome C. Okay, so it starts here and then it flows here, and after that it is not flowing. It's like coming back to this. So which is the same as here. From cytochrome C, it goes back to the center. Okay. So that is the electron flow through this reaction center in purple bacteria, having bacteriochlorophyll. So essentially this has 2 pairs, like 4 bacteriochlorophylls. One pair is in the reaction center, another one is the accessory pigment. So this structure was solved by these 3 scientists whose photograph and name are shown on the left. So this is where we have learned much of how the photochemical reaction centers work. And this is green sulphur bacteria, slightly more sophisticated than purple bacteria. What it does is, it does the same thing. An excited electron, the wavelength is different here. And this, from the excited reaction center, electron flows via quinone to cytochrome BC to cytochrome C back to the reaction center, generating a proton gradient. But at the same time, some of the electrons transfer to ferredoxin, a ion sulfur center. Remember, we learned about the ion sulfur centers in the mitochondrial oxygen electron transfer as well. From there to NAD reductase, ferredoxin NAD reductase, where you generate some reducing equivalent. Okay. And this could be used in dehydrogenation reactions to oxidize other molecules to obtain energy. So this is a little sophistication over the previous one. So this in the plant and cyanobacterial chloroplasts is equivalent to what we are going to call as reaction center type 1. And the previous one is equivalent to reaction center type 2. So both together we will see in chloroplast, cyanobacterial photosystem. Yeah. So this is the more complex Z scheme of the photosystem that operates in cyanobacteria and plants. So most of what we study come from the cyanobacterial photosystem. Okay, so that's less complex than the plants. So these two photosystem organisms like cyanobacteria, they use oxygen here. Okay, so this we did not see in the previous diagrams. In the previous diagram, from cytochrome BC1 complex, you get to cytochrome C back to the reaction center. Here that does not happen. Instead, it goes to the next system. So the two photosystems work in tandem. So we call this 2 first and 1 second. It is primarily because of the evolutionary history of characterizing this later and this first. So see, from one photosystem, it actually goes to the next photosystem. So electrons do not return. And this, as usual, pumps the proton, and therefore you get a proton gradient produced. And so therefore the electron hole is filled by electrons obtained by another really amazing molecule called the oxygen evolving complex. We will learn about it probably next class, which will be next Tuesday. So here the electrons are obtained by breaking water into oxygen, and so that is where we get the electrons, and that electron gets to the reaction center. So this is an important feature of the oxygen evolving photosynthesis or oxygenic photosynthesis. These use chlorophyll A and B. These do not use bacteria chlorophyll. And the purple bacteria and green sulfur bacteria, which did not evolve oxygen because they do not split water, they are called anoxygenic photosynthesis. So most of the time in our imagination, photosynthesis means we always associate with oxygen. It's not the case. That's only in oxygenic photosynthesis where water is the donor of electrons. And therefore, when you split water, you end up producing oxygen. The other ones do not. So here, the excited electron goes via pheophytin to these, plastoquinone A, plastoquinone B, and then to cytochrome BCF complex, which pumps proton, and then to plastocyanin, and from plastocyanin to the photosystem 1 reaction center. And this can also absorb light. So it gets excited by both the exciton transfer coming from this, in terms of the electron here, electrons coming from here, as well as the light absorbed here. So, and it reaches to the excited state, and from there, then via these accessory chlorophyll and then phylloquinone to ion sulfur to ferredoxin NADP reductase. Remember, this is like the structure we saw in green sulfur bacteria, photosystem 1. And this is more like the photosystem 2, purple bacteria. So that's why they have kept the same color theme here. Okay. So, and ends up producing a reducing equivalent. So here it is not NAD, instead it is NADP, and therefore it gets reduced to NADPH. So this is what gets accomplished. So the end result is this: you have two water molecules split to evolving one molecular oxygen. So that's the reason to satisfy the stoichiometry, we start with two water molecules, and therefore you get 4 electrons, and they go and reduce 2 NADP molecules to NADPH plus H plus. Okay, and the energy is provided by 8 photons absorbed by the antenna as a whole. So now to look at the structure and mechanism of these photosystem reaction centers. So photosystem II of cyanobacterium looks like this. This is a schematic. Okay, it's a really complex structural biology exercise, crystallizing these membrane proteins, solving the structures, and knowing where which amino acid side chain is. It's a Herculean task, and people have done this. So much of power of structural biology actually has been directed on these really fundamental biochemical protein systems in mitochondria and chloroplasts. So here, this portion we will study separately in the next class. And today what we are going to actually focus on the importance of tyrosine again. We'll see later. So this complex actually has reactions and has 3 identical structures, D1 and D2, and the D3 is not visible here. So this is just one portion of it. Like this, you have 3 complexes. This is the monomeric form that is shown here. Like this, you have 3 of them. So this monomeric form has got 2 subunits, D1 and D2. Subunit meaning one polypeptide chain. This one polypeptide chain, this is another polypeptide chain. Although these are identical, the electron flow happens via only one of them, as shown in this diagram here. So electron obtained from water. So I'll talk about this manganese ions importance in detail when we go there. And from there, the electron via the tyrosine residue gets to this chlorophyll molecules. Okay, this is the special chlorophyll at the reaction center. From there to pheophytin to quinone 1, then quinone 2 via this ion, and then from here it goes to cytochrome BC, this complex 3 equivalent here, and then to plastocyanin. Okay, that is this structure. And photosystem 1, again, the main point that I am not telling at every one of the systems is the transfer here is happening in solid state, requiring very, very little time. It is a matter of single-digit picoseconds. That is primarily because of the rigid orientation of these pigments and proteins, etc. They are very specific orientations such that the transfer happens nearly instantaneously. There is no chance for diffusion of molecules and therefore limitation by the diffusion rate. That does not come into play here. The whole thing happens in solid state. And so this is a cartoon view again of photosystem 1. Okay, the previous one is photosystem 2. So this is the one that has 3 identical complexes. Okay, and each complex has 11 proteins. So we are not going to look at the individual proteins here. So this has 3 subunits: subunit A, B, and C. And so this is the path of the exciton transfer. So these are the antenna chlorophyll molecules in their own respective light harvesting systems, okay, light harvesting complexes. So you have the proteins associated with them. Each one of this is equivalent to the structure. Okay, so I want to make sure that you remember the correct orientation and mapping within these systems. So that is what you have here. So to the reaction center, okay, and you get electron from plastocyanin also from photosystem II. And the excited one flow in this direction, finally to this ion, and then to this subunit C to the ferredoxin, and then ferredoxin NADPH oxidoreductase will transfer to NADP. So this is on the stroma side, the open internal space within the chloroplast. And this is the thylakoid membrane. So this is the inner side of the thylakoid discs. And this is the actual crystal structure of the trimer, looking from the thylakoid lumen, okay, from inside the thylakoid membrane. If you look on the surface of that membrane, this is how it is going to look like. So the gray structures are the protein subunits, and so they are basically there to hold these chlorophyll, the light harvesting chromophores, chlorophyll plus the accessory pigments in the correct orientation, and they are shown in color. Since all of them are not going to be readily visible when we have the protein subunits as well, this image shows the pigments without the polypeptide chains. Okay, so this is the ion sulfur center shown here. So these are the bacteria chlorophyll. Okay, so let me walk you through this slowly. So the monomer of this photosystem I with all the proteins omitted. Okay, so this is for one of them, and revealing the antenna and reaction center chlorophyll, like the green that you see, the dark magnesium, you need to zoom in and look at it. At this, you don't see the magnesium there in each of these chlorophyll. And the carotenoids are the yellow that you see around there. And then this is the ion sulfur of the reaction center. Okay. So this is the structure of the pigments alone. And they are all held in this rigid orientation by the polypeptide chains. And this is what we saw in a cartoon view here. And okay, so we will run out of time if I get into this detail.
So,
yeah, go ahead.
In the photosystem II of cyanobacterium, there's a P side and an N side. So, is there an electron gradient there also?
In the
photosystem II of cyanobacterium.
Right, yeah.
So there was a P side in the lumen and N side in the stroma. So there's a electron gradient there also. Because I
right, yeah. Here you have electron gradient. See here, this see in this big shaded red arrow indicates a proton gradient, and that is happening. See, I'll tell you with this, and then I'll. Okay, so I can draw, right? I keep forgetting that. So you have this thylakoid membrane. So this outside we will say this is stroma. Okay, so this whole structure is here on this. So the proton is pumped across this. And therefore you're going to create more protons here than here. So you're going to have a gradient. And then you have an ATP synthase pump through which when it comes, you are going to make ATP. Okay, so that happens. So there is a gradient generated. So the whole purpose of this is to generate gradient. It is not simply to reduce NADP. So that is what this bacteria does. So if you look at this one, the electron comes back to it. So the whole thing is in the membrane, and this is happening on the membrane. So here simply to avoid diffusion or dissipation of energy, these are in solid state and therefore quick transfer. And in this process, the electron has come back. So what is the work achieved? The work achieved is the pumping of the proton across. Here, of course, you don't have thylakoid. This is a prokaryote. So here, like the mitochondria equivalent we saw, so here it is the cell membrane acts as the barrier, and the protons are pumped across the membrane onto the periplasmic space between the bacterial cell wall and the bacterial cell membrane, and that acts like the thylakoid lumen. And from there it comes back to the cytoplasm, and that's when you may have the ATP synthesis happening in purple bacteria as well as in green sulfur bacteria. In addition, green sulfur bacteria can do this as well. And cyanobacteria is also bacteria, so it doesn't have organelle. So there again, it is the plasma membrane that acts as the barrier. Only in chloroplasts, what I drew here applies. Is this clear?
Yeah, thank you.
Okay, no problem. So any other questions?